All right, y'all. I hope this is recording. Are we recording? Oh my God. All right, I guess so. Today we're going to talk about Chapter 11, Control of Gene Expression. And yes, we did do Chapter 12 before Chapter 11. That's okay. So today's question, most cells express only a very small percentage of their genes. And as an analogy would be if you have a dictionary, I guess nobody really uses dictionaries anymore, but if you did, you wouldn't need to look at all the words. You're only interested in a few words all at different times, maybe on different pages, but still only a few words. And there are some words that you'll never need. And in the same way, all cells have the same genes, but different types of cells use different genes. So how does the cell turn genes on and off? First of all, it regulates protein synthesis, when and where proteins are made. And that intervention, that control, can occur during every part of the protein synthesis process. And today we're going to talk about the different ways that it can happen. Remember that genes code for proteins, and that each DNA molecule can have hundreds of different genes on it. This one here has 3 genes, just for reference. Each gene codes for a different protein, but not all of those proteins are always being used all the time. So for example, only the cells in your eyes use the gene for the eye color protein. So we say that in your eye cells, that gene is active, and the protein that makes your eyes a certain color is being expressed. So transcription and translation are happening for that gene. In your skin cells, the gene is not active, the protein is not being made, it's not being expressed, because your skin has no use for an eye color protein, and making one would just be a waste of energy. Sometimes genes are triggered by life events, and a good example would be that maybe you have the genes for schizophrenia, but you don't have schizophrenia, or maybe not yet. So why does a stressful event cause the cell to start using that schizophrenia DNA? Why does it trigger those genes? And today we will find out. Go ahead and watch this animation. I've indicated the questions as well as where you should write your answers on the diagram. There's also one more additional thing to know that you'll find in the animation. This will help you review for the lecture and also keep this whole thing around because it will help you study for the final. One thing the animation doesn't mention that you do need to know is that during RNA processing, right here, between these two steps, RNA processing, 2 things happen. First, a 5' cap and a poly A tail are added to the mRNA, and that keeps it stable and protects it as it goes into the cytoplasm. And I've written that here for you. The other thing is the one the animation describes, so you'll have to write that for yourself. So go watch the animation and come back when you're done. All right, today we're talking about control. The cell can control which proteins it's made, when they make them, and where they make them by controlling protein synthesis. We're going to touch on a few major points. The first point of control is DNA packaging. Cells can control how their DNA is packaged. And there's a whole field of study devoted to this that's called epigenetics. Hey, no. They can scratch all over it. Anyway, it's called epigenetics, and it's about how your lifestyle choices influence how your DNA is packaged and therefore which proteins are made. We're going to spend some time on that. Other points of control include transcription, mRNA processing, and translation, but we won't talk about those. I just want you to know that the cell can control protein synthesis at these points, which is why I put them on there. We're also going to discuss how your body controls where all the different body parts go so you don't look like a Picasso painting. And we'll talk about how cells control other cells. We'll also talk a little bit about cancer. So, cells can control what proteins are made when and where by controlling how DNA is packaged. Watch this animation and answer these questions and come back when you're done. And now watch the textbook animation, which I have linked in the module, and answer these questions and come back when you're done. All right, So this is the way a cell organizes its DNA. First, it wraps the DNA. Well, hopefully you've caught that DNA is completely and fully packaged and wound up tightly into a chromosome only during cell division. But even when the cell isn't dividing, it's got to keep some of that DNA wound up tightly. There's just not enough room for it all to be stuffed in there without being tightly packaged up. Remember, there's 6 feet of DNA in each human cell, and you can put more stuff into the same amount of space if you fold it up tightly and tuck it away. Like if you try and stuff a bunch of unfolded underwear in a drawer, versus if you fold and roll each pair up into a little ball, then you can fit more underwear in your drawer if you do it that way. So here's how a cell organizes its DNA. First, it wraps the DNA tightly around these little proteins, 8 of these little proteins. They're called histones. And the resulting structure is called a nucleosome. And if you were to look at the piece of DNA through a very, very powerful microscope at this stage, it would look like beads on a string. Each individual bead represents, well, not represents, is a nucleosome. And then the nucleosomes start to stack on top of each other. And at this stage, the DNA is called chromatin. And when I refer to chromatin, what I mean is just DNA that has been wrapped around histones, has been wrapped around itself. Everything is all stacked up. Watch this animation, answer the questions, and come back when you're done. So how do we get the DNA to be accessible? Well, the first step of protein synthesis, remember, is for RNA polymerase to latch onto the DNA and then read the information in the DNA and create some mRNA. But it can't do that if the DNA is tightly wound around these histone proteins because there's no room for it to get in there. The histones block the RNA polymerase. And not only is the DNA tightly wound into these nucleosome structures, but the nucleosomes are also all squashed up on each other like this. And that means there's even less room for an RNA polymerase to get in there. So for RNA polymerase to be able to access the gene, a couple of things have to happen. First, nucleosomes have to separate a little. They have to go from being so tightly packed together to being more loosely associated, like this. Once that's done, we've got to loosen up the DNA from around the histones, right there, and make some room for RNA polymerase to come in and do its job. The whole thing is about making room for RNA polymerase to access the DNA. So let's watch this animation together. Right. That's to mute it. Come on, computer, you got this. All right, so here we've got our chromosome. We've got the DNA all wound up. There's the chromatin. And there's the nucleosomes. Each nucleosome is a complex of 8 histone proteins with DNA wound around it. And the nucleosomes have little tails. You can kind of see them. Right? Those are the little. There are enzymes that carry these chemical groups called acetyl groups, and they attach them to the tails of the histones, like that. And as they do this, the DNA loosens from around the histones. So now there's room for RNA polymerase to get there and start making some mRNA. See, it can in there. And when the acetyl groups are attached to the histones, they also sort of unwind the chromatin and separates the nucleosomes from each other. So there's room for RNA polymerase in there. And histone, there's room for RNA polymerase here between the histones. So not only can the RNA polymerase fit in and access the DNA around the histones, but it can also access the DNA between the histones. All right, I want to clarify something on this picture. Each of these little cylinders represents a nucleosome, each of these little guys. And remember, a nucleosome is a group of 8 histones with some DNA wound around it. So in the larger picture, they haven't gone to the trouble to draw each of these 8 histone proteins. They've just simplified it. But each of these cylinders is not an individual histone. It's a group of 8 histones, that is, a nucleosome. So if we take a look at this picture, we can see that we've gone from this to this. We've gone from this to this just by adding some acetyl groups to the histones. When the chromatin is tightly wound up, it's not accessible to the DNA, to the RNA polymerase, I'm sorry, is DNA. When it's not accessible to the RNA polymerase, then we call that form heterochromatin. And once we've added some acetyl groups and the chromatin is loosely wound and the DNA is accessible, we call that form euchromatin. And here you see that RNA polymerase is busy transcribing the DNA. It's reading the DNA now that it can access it, and it's making the corresponding mRNA. And you can remember which one is which by the prefix U, EU. This means good. So the chromatin that's open, the euchromatin, that's the quote-unquote good chromatin. That's the one that can be used. And hetero means other, so the heterochromatin is the other chromatin. This is what an acetyl group actually looks like. It's an oxygen with a double bond to a carbon, because remember we assume a carbon at each corner. And then there's another carbon and 3 hydrogens over here. Changing the slide. All right, let's summarize. In the picture, each of these little balls represents a nucleosome. The process of adding acetyl groups is called histone acetylation. Histone acetylation causes the nucleosomes, these guys, groups of histones, to separate and the DNA to loosen its grip around the histones. When the chromatin is in this open form, we refer to it as euchromatin. In euchromatin, there's room for RNA polymerase to fit in between the nucleosomes and mRNA can be made, which is the first step of protein synthesis. So let's add some little acetyl groups here. Oh yes, histone acetylation allows transcription. So let's add the little acetyl groups right here. Each of these little dots represents an acetyl group. So you can see nothing is happening to the DNA. The DNA remains the same. We're just modifying the histones. And when the acetyl groups are removed, the chromatin closes up and it goes into its heterochromatin form. So the RNA polymerase can't attach to the DNA and transcription won't happen. Let's watch this animation, the rest of this animation together. I'll mute that as well. You can do it, computer. I have faith in you. All right, so there's the DNA getting loose, all loosified. And then we're going to take the acetyl groups off. And it just winds the chromatin back up. Now the RNA polymerase can't get in there. And that is heterochromatin. There we go. All right, so go ahead and do this check yourself question. The answers I'm going to give you at the end of the PowerPoint. All right, well, there's a lot of genes that you don't want to be transcribed in certain cells. For example, your skin cells should not be making blue-eye protein. And one way cells control this is by adding methyl groups to the DNA. Note that acetylation added things to the histones, but methylation adds things to the DNA. It doesn't change the DNA, it just sticks these methyl groups onto the DNA. And when the methyl groups are attached to the DNA, it automatically winds up into this structure of heterochromatin. And in heterochromatin, there's no room for RNA polymerase to get into the DNA, so transcription can't happen. So let's add some methyl groups to this DNA over here. The methyl groups will be red. And notice we're adding it to the DNA, not to the histones, just to the DNA. There we go. DNA methylation prevents transcription, and histone acetylation promotes transcription. All right, do this check yourself question. I'd like to make a little note of something. Histone acetylation and DNA methylation don't change the DNA sequence. They are reversible under different conditions. In DNA methylation, the little methyl tags are just stuck onto the outside of the DNA. It doesn't change the DNA. Changes to the DNA sequence, the order of the nucleotides, the A's, C's, G's, and T's, those are permanent. But DNA methylation and histone acetylation aren't permanent, they're reversible. Your actions, your mindset sometimes, if that stresses you or not, lifestyle, all of these things can either cause or reverse DNA methylation. And even though they don't change the DNA itself, they can still be passed on to future generations. So the things that you do to affect your methylation can potentially affect your kids, your grandkids, etc. Correct methylation and acetylation are really important. For example, insufficient DNA methylation can lead to abnormal embryonic development. And the study of all this is called epigenetics, the study of inheritance of traits transmitted by mechanisms not directly involving the DNA sequence. Whew. Excuse me. All right. Watch this animation. I do want to clarify one thing. So one thing I don't like about this animation is that it represents a nucleosome like this and makes it look like there's only one histone. But just remember that there are 8 histones per nucleosome. All right. So we're done with DNA packaging. Now let's move on to epigenetics. So we know that proteins are made based on DNA, and we know that DNA is made accessible or not by whether it's acetylated, methylated, or neither, and to what degree. We know that this pattern of acetylation and methylation and the resulting expression of proteins is called the epigenome. And your activities and your lifestyle affect your genome. That is, which parts of your DNA are accessible and which parts are not. We just discussed that DNA methylation doesn't change the sequence, it can be reversed. All the same, these patterns can be passed on to your kids and even your grandkids. Epigenetics is the study of the inheritance of traits transmitted by mechanisms not directly involving the DNA sequence. And in the field of epigenetics, the acetyl and methyl groups are called the epigenetic tags. Sometimes they're called epigenetic signals. Okay, answer this question. Go ahead and watch the epigenome at a glance and answer that question. And then do the same for this. Answer those questions. All right. Insights from identical twins. Twins start out with identical placement of epigenetic tags, meaning methyl groups. Their epigenomes are identical, but as they grow older, their epigenomes become more and more different from one another. Here is a pair of chromosomes from a pair of twins. The twin's epigenetic tags are dyed red, and the other are dyed green, and wherever you see yellow, there's an overlap of them. This is chromosome 3, and the pairs of chromosomes have been layered on top of each other. When the twins are 3 years old, there's a lot of yellow, so you can tell that there's a lot of overlap. Many areas of these chromosomes are identical between the two children. However, as they grow older, each individual makes specific lifestyle choices, has different experiences, and as these things happen, the epigenetic tags, that is the methyl groups, are removed from areas of DNA, they're added to others, and by the time they're 50, the epigenomes are very, very different. You can see that there's hardly any yellow, hardly any overlap between the two. The DNA itself is still the same, but the pattern of methylation is different. Nearly the entire thing shows differences, which you can see with the red and green. Epigenome learns from its experiences. Epigenetic tags act as a kind of cellular memory, and each cell has a different epigenetic profile. A cell's epigenetic profile is the sum of the signals it has received during its lifetime. It's a collection of tags that tell genes whether to turn on or turn off. As the epigenome changes, gene expression changes. Just after fertilization, an early embryo is made up of stem cells, and it'll divide a few times. So the first few cells are stem cells, and stem cell is a cell that can become any other kind of mature cell as it matures. So any of these cells could, where's my mouse? Come on. Any of these cells could become a bone cell, a muscle cell, a nerve cell, any kind of cell in the human body. But as the cells continue to divide, they become different from one another. Different sets of genes are activated in different cells, and this makes them differentiated cells. A differentiated cell is a cell type of a specific type. Basically, a cell that's not a stem cell. It's been specialized for a specific function. And once the cell has been differentiated, once those different sets of genes have been activated or inactivated, in the end, only 10 to 20% of all the genes are active in any given cell. And whether the genes are active, excuse me, determines what the cell will be. For example, both muscles and nerve cells have the same genes, but only the muscle cells use the genes that are needed for muscle, and the brain cells only use the genes that is necessary for brain function. Another check yourself question. Right, and then also environmental signals. As the environment changes, our epigenome changes. And as the epigenome changes, which genes are expressed, that is, the ones are turned on and off, those change too. The advantage of all this is that our genes allow us to adapt to our specific environment. As DNA is replicated, the placement of methyl group stays the same on the new DNA strand as on the original DNA strand. Using the original DNA strand as a template, these special enzymes called methyl copying enzymes attach methyl tags to the new DNA strand so that they're identical to the original DNA molecule. There was a study done on the effect of maternal nurturing in rats. One mother rat was just a super relaxed rat. She was very mellow, spent a lot of time licking and nurturing her babies. And these baby rats turned out to be pretty relaxed rats themselves. Another mother rat, was very anxious and stressed. She didn't take a lot of time to lick and nurture her babies. And those baby rats turned out to be very anxious themselves. And we know that this is not genetics, because whether the pup grows up to be anxious or relaxed depends on the mother that raises it, not the mother that gives birth to it. And the researchers found that different sets of genes were active in the stressed pups versus in the relaxed pups. This shows that the epigenome changes with an organism's environment. This adaptation of the epigenome is actually really important because it prepares the pups for a future environment. Anxious, guarded behavior of the low-nurtured rat has an advantage if it lives in an environment with high stress and where food is scarce. This rat is likely to keep a low profile and respond quickly to stress. But if you were to put the relaxed rat in this same high-stressed, uncertain environment, that relaxed rat would probably be eaten by a predator pretty quickly because it would tend to let down its guard. And of course, eventually the pups grew up, and then researchers observed how each one raised her young. The ones that had received high quality care became high nurturing mothers, and the ones that received low quality care became low nurturing mothers. Behavior itself affected the epigenome. The nurturing behavior transmitted itself onto the pup's DNA. So all this provides evidence that parents play a huge role in shaping the epigenome of their offspring. Your environment, your lifestyle choices, they will affect your children and even their children. But epigenetics in humans is really difficult to study for a few reasons. First of all, humans have a very long lifespan as opposed to rats or other animals, which means that any experiment done on a human could take 70 to 90 years. And in addition to that time for the first generation, whatever study happens must span several generations, because if you're studying an individual who becomes a mother, not only are you studying her, but you're also studying her child and her child's children through the reproductive cells of her baby. So you're talking about an even longer time of study. The epigenome can change very quickly. So if you take some DNA from a person in 1 month, the epigenetic tags might be slightly different if you sample their DNA the next month. And last, because the epigenome is temporary and it's constantly changing, an epigenetic change that was triggered by certain environmental conditions may be reversed when those conditions change back again. In a new embryo, most of the epigenome is erased at the beginning. Most of the methyl tags that activate or silence certain genes are removed. This allows the cells to have a genetic fresh start or a blank slate, sort of. This process is called reprogramming. It resets the epigenome of the early embryo so that the initial cells are once again stem cells. They can form every type of cell in the body. In order to pass to the next generation, epigenetic tags must avoid being erased during this reprogramming process, and some of them do. For a very small number of genes, epigenetic tags make it through this process and they pass completely unchanged from parent to offspring. That's called imprinting. In mammals, about 1% of genes escape epigenetic reprogramming and become imprinted genes. And whatever tags become imprinted, those are preserved in your children. So whatever choices you make, environment you live in, any epigenetic tags from those choices have a chance of being passed on to your children because you can't control which ones are going to be imprinted and which ones are not. Go ahead and answer this check yourself question. And now we're moving on to homeotic genes. Thank God nobody actually looks like a Picasso painting, but why not? Here's why. Your body knows how to put its parts, where to put its parts by using a set of genes called homeobox genes. They're also called selector genes. These are master regulator genes that quote unquote select the identity of entire segments or entire structures in organisms. They tell your arms to go on your torso instead of coming out of your butt, for example. If these genes are mutated, if they're inactivated, that could cause body structures to develop in the wrong place, or if they're overactive, that too. The homeotic genes are also called Hox genes. Most of the homeobox genes code for proteins that don't necessarily make body structures, but they control the activity of the other genes that do make the body structures. So for example, in your eyes, the homeobox genes are making proteins that silence the gene for arms. This is a fruit fly. A species is Drosophila melanogaster. Fruit flies are really useful for experiments because they're cheap to work with and they have a really short lifespan. And here you can see a portion of one of the fruit fly chromosomes. Scientists have mapped the locations of these homeotic genes within the chromosome. And they have also mapped which gene controls which body segment. You can see here that the lab gene controls the placement of the mouth parts, the PB gene controls the front of the head, and so on and so forth. Here's a great example of what happens if a Hox gene is mutated or turned on when it shouldn't be. Scientists have been able to get the fly's body to turn on the antennapedia gene in the head of the fly instead of just the torso in the head. And this is what happened. Well, on the left is a normal fruit fly, and on the right is a fruit fly whose antennapedia gene has been activated in its head. And instead of antenna, there are legs where the antenna should be. Homeobox genes are messed up, so the body part placement is messed up. That's all of that. Let's talk about cell signaling and signal transduction pathways. A signal transduction pathway is just a set of chemical reactions in a cell that occurs when a signaling molecule attaches to a receptor on the cell membrane. Basically, these allow cells to regulate each other's gene expression. If one cell sends out a protein into the bloodstream or into the tissue, that protein acts as a signal when it attaches to another cell, the surface of another cell, then that's going to cause a cascade of events that will affect gene expression in the target cell. So here's the steps of cell reception, cell signaling. A signal molecule is sent out by a cell, any cell, sends out a protein, and the protein signal will bind to the receptor on the surface of the target cell. This activates a series of proteins within the cell, which in turn activate the other proteins and activates another protein, and so on and so forth. And eventually it produces a cellular response of some kind. It's a lot like a chain of dominoes. If you push down the first one, then all the others just fall over until the end. And the cell that sends out the signal molecule is controlling the actions of the target cell. So watch this animation and find out what a transcription factor is and what it does. Cell division is regulated by signal transduction pathways. It's regulated by proteins that tell neighboring cells when to stop dividing. And if cell division just keeps going and going and going, that forms a tumor, a mass of cells that doesn't stop growing. Here's some vocabulary that you need. An oncogene is what we call a gene that causes cancer, and a proto-oncogene is a normal healthy gene that if it's mutated, it could become an oncogene and cause cancer. Many proto-oncogenes produce proteins that regulate cell division through signal transduction pathways. And if these genes are mutated, cell division will go unchecked. It will just keep dividing and dividing and dividing. Then we've got our tumor. So there's two proto-oncogenes involved in regulating cell division. They're called RAS and P53. And you can think of RAS as being the gas pedal, the go signal for cell division, and P53 as being the stop signal or the brakes for cell division. And if there's mutations in either of these genes, that's like having car problems where there's too much acceleration or the brakes don't work. And in either situation, you're probably going to have an accident. But a mutation in either of these genes, these proto-oncogenes, will cause it to become an oncogene and cause cancer. And here is how. So the RAS gene makes the RAS protein, and that encourages cell division. And think of it like the gas pedal of cell division again. RAS is a key part of a signal transduction pathway. So in a normal cell, a growth factor released from another cell will bind with the target on this cell's surface. That's a protein target. And when the growth factor binds, it causes the receptor protein to change shape. And that causes a series of chemical reactions. The signals are transferred from one protein to the next, and the next, and the next, and the next. And finally, transcription and translation happen, which makes a protein that causes the cells to divide. Note that the cell that sent out the growth factor is controlling, essentially, cell division of this cell. The product of the RAS gene is a key component in this signal transduction pathway. There's a mutation in the RAS gene, then suddenly it becomes a gene that can cause cancer. It becomes an oncogene. The mutation will produce an overactive RAS protein, and suddenly it's like someone has put a brick on the gas pedal. This protein will cause cell division even if there isn't any growth factor. We have increased cell division during a time when the cell is not supposed to be dividing. The cell just keeps dividing and dividing and dividing, and that's what a tumor is. It's just a mass of cells that simply cannot stop growing. All right, now let's talk about P53. Remember, this is like the brakes of cell division. It prevents the cell from dividing. And normally, if the body wants to keep a cell from dividing, it will send that cell a protein called a growth inhibiting factor. The growth inhibiting factor binds to a receptor on the surface of the target cell, and that causes a chain of events signaled through proteins activating one another, leading to creation of a protein that stops cell division. So the P53 protein is part of this signal transduction pathway. It allows activation of the gene, and then that makes a protein that inhibits cell division. So there's no cell division. This P53 protein, yeah. And note that the cell that sent out the growth inhibiting factor is controlling the actions of the target cell. If there's a mutation in the P53 gene, then we get a non-functional P53 protein. So all of this signaling still occurs, but transcription and translation can't happen because there's no P53 protein. And that means there's no division repressing protein being made. And so cell division just keeps going and going and going. The end. And here are the answers to the check yourself questions. First, histone acetylation allows increased transcription. Second, DNA methylation closes up chromatin structure. Third, your epigenome is your specific pattern of methylation and acetylation and the resulting protein synthesis. A stem cell is a cell that can become any other kind of cell. It's good that the epigenome is flexible because it allows your body to adapt to your environment by by controlling which proteins your body makes. Oh, that's the end. So, yep. Bye.